Go.
This is Unit 4 of AP Bio. So we start over here in the left corner. You have the types of cell signaling. First off, you have juxtacrine, which is right here. These cells are touching. They're just like doing a handoff. Next, you have synaptic, which comes all the way over here. These are two cells in the synapse sending neurotransmitters as a way of cell signaling. This is endocrine. It's just two cells. They're not touching, but they're close to each other. The cell is just passing the ligand over. And then you have local, which is just, again, going from one part of a cell to the same part of the same cell who sent it. Next off, you got the signal transduction. This is where the cell signaling is being received. First, you have a receptor, which is this part, and the ligands are like very specifically binding to it, and that sends the signal inward, only if the kinase is allowed, because this needs to be phosphorylated for it to be activated. So the kinases make sure this is phosphorylated so that the signal transduction pathway is active, things are coming through. If the protein phosphorylates didn't want that, they would dephosphorylate this, which shuts it completely off. Anyway, so the signal gets into the cell, it gets phosphorylated, and it gets turned off from on. And as more phosphorylated, it gets amplified by secondary messengers like CAMP. And then finally, it reaches the nucleus, where it can derive a response. Responses include turning a gene on or off, which just means whether or not it's going to be produced. Membrane permeability, how much stuff is going to get in or out of that membrane. And then altering the metabolic process, which is alters your metabolism, so enzymes. And now we're here at feedback loops. So you have negative feedback, which decreases the stimulus and brings you back to homeostasis. So for example, you're sweating, the stimulus is that you're really cold. You're really hot, so you're sweating, and the response for that is another response. My bad. What is it?
It's decreasing temperature.
Yeah, decrease in temperature, and so it's gonna bring you back to homeostasis. It's gonna decrease the stimulus. Positive is the inverse. It increases the stimulus, so it increases the responses. Some examples include blood clots and child labor for women. You can't be in homeostasis when you're disease, sick, or have a virus. And over here, you have another example. So you have stimulus, receptor, effector, and response. This is what I meant earlier. The stimulus is that you're sweating. The receptor is your temperature receptors in your skin. The effector is that you're going to sweat. And the response is that you'll cool off. Another example is the glucose levels in the blood. When there's a lot of glucose after you just ate a meal, that's the stimulus. The receptor is like the body's going to detect the glucose in the blood. The effector is insulin's going to be sent out. The response is going to be lowered glucose levels in the bloodstream. And now we're on to mitosis. So to start, we need to see how the chromosomes are made. DNA is going to start out by wrapping around a protein called a histone. Once it wraps around the histone, it becomes a nucleosome. And these are just strands of whatever. But then when the strands of the nucleosomes condense, they make chromatins. Not when they condense, they just make chromatins. They combine to form chromatins. When chromatins condense after DNA replication, they make a chromosome. And so to begin mitosis, we need to start with interphase, which has three phases in itself. G1, which is for cell growth and regular functions. And then you have the S phase, which is for DNA and chromosome replication and duplication. And then finally you have G2, the final growth, and it's for preparation for mitosis. Oh, my bad. No, that's not the final one. You also have the M phase, which is mitosis and cytokinesis down here, but we'll cover that in a second. There's different checkpoints in this phase. The first checkpoint is G1, and basically what G1 detects for is the cell size, any growth factors, and DNA damage. If it detects anything it doesn't like, it stops. It sends the cell to G0, and this cell will no longer divide, and it can potentially return to G1 phase, but it probably won't. It's usually permanent. It goes to become like a muscle or nerve cell. If it gets the green light and it's good, it goes to complete the whole cell cycle. Down here you got the G2 checkpoint. Pretty similarly, it detects for DNA damage, but it also detects for DNA replication or poor DNA replication. If it's bad, the cycle stops and it just pauses and lets the cell try to fix itself. If the cell can't do that, it does apoptosis, regulated cell death. The cell's no more, it's dead. If it gets the green light, it goes on to mitosis down here in this checkpoint. Mitosis, it detects for microtubule attachment to chromosomes and kinetochores. So, if it detects a red light, it says don't go. It pauses mitosis and it allows the spindles to finish attaching to the kinetochores. If it's already good, it gets the green light and it goes to anaphase, over here, and it finishes mitosis. Now we gotta understand all the phases. First up is prophase. So you have the nuclear envelope here, and some of like the spindles are starting to form. Then you go to prometaphase. This like bursts and fragments, and things start to line up down here. Next, you got the metaphase, where they line at opposite poles, and the DNA strands dead center. And then here, the spindle fibers, they connect to the kinetochores. Is it the spindle fibers or microtubules?
It's like the same thing.
The same thing,
Yeah.
anyway. It connects to the kinetochores, like the middle parts of the chromosomes, and it pulls them apart. And so now they're separated at the opposite poles. And now they're in their own little clusters. Telophase starts to form a nuclear envelope around the cluster here and here. And then cytokinesis finishes by splitting the cell, separating the two clusters, and making own new cells. So now you have two cells that look similar to this, where they have the nuclear envelope, and then they're all alone.
Then also just one little thing, this area for metaphase is just called the metaphase plate.
Oh yeah, the metaphase plate.
Yeah.
And that's it.